Hello students, welcome to readmedprep Academy channel. Today we are going to talk about the chromosomal basis of inheritance part 4, in which we will discuss multiple alleles, characteristics of multiple alleles, self-sterility in Nicotiana and sex determination in Maize. What are multiple alleles? A given phenotypic trait of an individual depends on a single pair of genes, each of which occupies a specific position or locus on the homologous chromosome. When any of the three or more allelic forms of a gene occupy the same locus in a given pair of homologous chromosomes, they are said to be called multiple alleles. There may be multiple alleles within the population, but individuals have only two of those alleles. What are the characteristics of multiple alleles? Multiple alleles of a series always occupy the same locus in the homologous chromosome. Therefore, no crossing over occurs within the alleles of a series. Multiple alleles are always responsible for the same character. The wild type alleles of a series exhibit dominant character, whereas the mutant type will influence the dominance or an intermediate phenotypic effect. When any two of the mutant multiple alleles are crossed, the phenotype is always mutant type and not the wild type. Now, let us discuss the self-sterility in Nicotiana. In plants, multiple alleles have been reported in association with self-sterility or self-incompatibility. Self-sterility means that the pollen from a plant is unable to germinate on its own stigma and will not be able to bring about fertilization in the ovules of the same plant. It is a genetic mechanism which prevents self-fertilization, thus encourage outcross. East in 1925 observed multiple alleles in Nicotiana, which are responsible for self-incompatibility or self-sterility. The gene for self-incompatibility can be designated as capital S, which has allelic series S1, S2, S3, S4 and S5. The cross-fertilizing tobacco plants were not always homozygous as S1, S1 or S2, S2, but all plants were heterozygous as S1, S2, S3, S4, S5, S6. When crosses were made between different S1, S2 plants, the pollen tube did not develop normally, but effective pollen tube development was observed when crossing was made with other than S1, S2, for example S3, S4. When crosses were made between seed parents with S1, S2 and pollen parents with S2, S3, two kinds of pollen tube were distinguished. Pollen grains carrying S2 were not effective, but the pollen grains carrying S3 were capable of fertilization. Thus, from the cross S1, S2 is to S3, S4, all the pollen were effective and four kinds of progeny resulted. The progeny is S1, S3, S1, S4, S2, S3 and S2, S4. Here you can see in this image the self-incompatibility in relation to genotype in tobacco. The stigma is S1, S2. When the pollen grain of S3, S4 falls on the stigma, the S3, S4 pollen grain grows its pollen tube into the stigma. When a S3, S1 pollen grain falls on the stigma of S1, S2, S1 does not germinate, whereas S3 pollen tube enters into the stigma. When a S1, S2 stigma is taken and S2, S1 pollen grain falls on the stigma, the S2 or S1 does not germinate on the surface of the stigma and grows into the pollen tube. So, different combinations of progeny in self-incompatibility. You take the female parent on one side and the male parent. Male parent may be S1, S2 or S2, S3 or S3, S4, which is the pollen source. When you take the stigma of S1, S2 and the male pollen source as S1, S2, the self-sterile occurs. When you take the stigma of S1, S2 and the pollen of S2, S3, the combination is S3, S2 and S3, S1. When you take the stigma of S1, S2 and the pollen source as S3, S4, the combination is S3, S1, S3, S2, S4, S1 and S4, S2. When you take the stigma of S2, S3 plant and the male pollen as S1, S2, the combination is S1, S2 and S1, S3. When the stigma is S2S3 as well as the pollen source is also S2S3, it is self-sterile. Whereas if you take the stigma of S2S3 and the pollen source as S3S4, the combination is S4S2 and S4S3. If you take the stigma as S3S4 and the pollen source as S1S2, the combination is S1S3, S1S4, S2S3 and S2S4. When you take the stigma of S3S4 and the pollen source as S2S3, the combination is S2S3 and S2S4. When you take the stigma of S3S4 and the pollen source as S3S4, then there will be no germination of the pollen grains and it is self-sterile. Now let us discuss the sex determination in maize. Maize is monoecious. Z maize is an example for monoecious, which means male and female flowers are present on the same plant. There are two types of inflorescence. The terminal inflorescence, which bears the staminate flower. The staminate florets develops from shoot apical meristem called tassel. The lateral inflorescence, which develops pistillate florets from the axillary bud, is called the ear or cob. Here you can see the inflorescence of Z maize, the terminal inflorescence which has the staminate florets called the tassel, and the ear, which is the lateral florets or the axillary florets which have the pistillate florets. Unisexuality in maize occurs through the selective abortion of stamens in the ear florets and pistils in the tassel florets. So what is feminizing and masculinizing? Feminizing means to induce female characters in a male. Masculinizing means to induce male characters in the female. Now let us discuss about this. Gene pairs small b, small a and small t, small s. A substitution of two single gene pairs, BA for barren plant and TS for tassel seed, makes the difference between the monoecious and the dioecious, which is rare. Dioecious is rare in the maize plants. The allele for barren plant, BA, when homozygous, makes the stalk staminate by eliminating silk and ES. The allele for tassel seed, TS, transforms the tassel into a pistillate structure that produces no pollen. So the table in the next slide is the resultant sex expression based on the combination of these alleles. Most of these mutations are shown to be defects in the gibberellin biosynthesis. Gibberellins play an important role in the suppression of stamens in florets or ears. Here you can see in this table the sex determination in maize. The superscript plus denotes the dominant character. Here in the table you can see the genotype, the dominant or recessive, what is the modification and what is the sex of the plant. BA, BA, TS, TS. It is a double recessive. What is the modification? It lacks silk on the stalk but transformed tassel to the pistil. So the sex is rudimentary female. BA, BA and TS plus, TS plus. It's recessive and dominant. Lacks silk and have tassel, so it's a male sex. BA plus, TS plus, TS plus. It's a double dominant. Have both tassel and cob. This plant is monoecious. BA plus, TS, TS. It's a dominant and recessive. Bears cob and lacks tassel. It's a normal female. So today in chromosomal basis of inheritance, part 4, we have discussed multiple alleles, the characteristics of multiple alleles, and how self-sterility occurs in Nicotiana, and something about the sex determination in maize. Thank you. Kindly subscribe, like, share and comment to channel Read MedPrep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much, students.